Hello and welcome, this is Alex from Phuket Pals with another GD Life at Pals lesson on GD Science, and today we will learn about the topic heredity, or more precisely, inheritance. Let's dive right in. So we will start with DNA and chromosomes. If you want to know about these things in more detail, there are other online lessons and screencasts where you can learn more about this topic. We will just have a little overview and review here about this part again. So a cell uses the instructions in its DNA to make substances called proteins. And proteins are responsible for producing all our physical traits, such as your eye color, your hair color, even your height to some extent. Anything you look at your body basically is based, all these traits are coming from your DNA. And a gene is a segment of DNA, a specific length of DNA that codes for this specific trait or a specific protein. So if we look at the following diagram of DNA, DNA is made up of 4 DNA bases or chemical bases, which are called adenine, thymine, guanine, and cytosine. Or more simply put, we often see just the letters A, T, G, and C, where A always goes together with T and G always goes together with C. Our DNA is a double strand where these bases pair up, and these sequences of bases, so one base coming after the next, this can code for a specific protein, for a gene. We will have a look at this in more detail now. So the process of using a gene to make a protein happens in 2 major steps. These are shown in the graphic down here. This is one of the central dogmas of molecular biology. First, the gene, a certain length of DNA, is transcribed, the process of transcription. To transcribe simply means to copy. So DNA, a certain piece of DNA, a part of DNA, length of DNA, the gene is copied or transcribed into a molecule that's very similar to DNA, which is called RNA. And the RNA is simply a copy of that specific gene and contains the information for that gene. This RNA is then carried out of the nucleus, where the DNA is located, and is brought to a so-called ribosome. Again, if you are not sure what these things are, the nucleus, ribosomes, there is another lesson on these cell structures where they are explained in detail. The RNA goes to the ribosome, and the ribosomes are like small factories that are able to read the information that is located now in the RNA and use that information to build a specific protein. This process of building the protein is called translation. So the information that is stored in the RNA coming from the gene is then translated or used as like an instruction manual to build the protein from amino acids. And these specific proteins then lead to traits in the organism. As you know, children typically exhibit a mixture of traits from both of their parents. You might have, for example, your mother's eye color and your father's face shape. So when people and other organisms reproduce, they pass on these traits to their children through their DNA. And during reproduction, DNA is passed on to the offspring of the parents. How that exactly works, we will see here. So first let's have a look at chromosomes. What are chromosomes? Again, more detail in another lesson if you're interested in that and need to revise on that topic a bit more. So all our body cells contain DNA, and as well our reproductive cells, the sperm cells and the egg cells. And the DNA forms structures called chromosomes. So a chromosome is a long molecule of DNA wrapped around specific proteins. So when an egg and a sperm cell unite, they create a new organism. This first cell is called a zygote. So here we have a sperm cell and an egg cell. When they combine, this is the process of fertilization, and we get a zygote, the first cell of the new organism that has the chromosomes of the mother and the chromosomes of the father. Father's chromosomes coming from the sperm cell and mother's chromosomes coming from the egg cell. So half of the mother's chromosome and half of the father's chromosome come together. We can see here that before sperm cells are made, specific cells have to go through a process called meiosis, a certain type of cell division. Again, more detailed description in another screencast. And meiosis is a so-called reduction division, where the number of chromosomes from the father's and mother's cells is halved, so that sperm cells and egg cells have half the number of chromosomes. No, same is true for the egg cell. Egg cell has half the number of chromosomes. These two cells unite, and we know that a half plus a half is 1. So the new cell has one complete set of chromosomes again. That's very important, that the number of chromosomes is correct. That cell will then divide and pass on the correct number of chromosomes to all the other cells of the new organism. The chromosomes that store DNA are located inside the cell's nucleus. Now we can see a chromosome here. It consists of 2 identical chromatids, and these are attached to each other in the center at the so-called centromere. And a single chromosome can contain hundreds or even thousands of genes. And each single gene is responsible for a specific protein, which will be responsible for a specific trait. These genes can be, again, read, transcribed, and copied into mRNA, and that can be translated at the ribosomes into a protein. All right, a new term, alleles. Alleles and mutations. Alleles are different versions of a specific gene. So each gene can have different types of alleles. Most genes have 2 alleles, though more alleles are possible for a single gene. For example, one of the human eye color genes has 2 alleles, brown color and blue color. So we have the gene for eye color, and we have the 2 alleles, brown eyes and So brown eyes, blue eyes are the two alleles for eye color, the two versions of the gene for eye color. The human blood type, for example, has three alleles, A, B, and O. New alleles for a gene are created by mutations. A mutation is a random change in the base sequence of a gene, and the following diagram here on the right shows different types of mutations that can occur and lead to new alleles. And we have to say here that most mutations that happen are usually mutations that are not beneficial. Sometimes a mutation can be beneficial that leads to a more beneficial allele that can give an advantage to an individual. If a mutation occurs, it is very important that if it is a new allele that can be inherited or passed on to the offspring, then this mutation has as well to be in the sex cells, in the gametes and the egg or sperm cells. If they just happen in a random body cell, then the individual will have that new allele, but it's not able to pass it on to the next generation. So here we see the normal gene with its base sequence. We've learned we have the four bases, C, A, G, and T. We have a certain sequence here, and this sequence can mutate in different ways. We could exchange a single base, so this becomes a C. Before we can see it was a T here, now it is a C. This would be a single base change, mutation. We could have an addition of a new base. So we are adding a new base in. Now we can see from here to here, we have a C here and a C here, and we've just added in another C. Or we can have deletions. Certain parts of the DNA sequence can be deleted. And all of these will lead to changes in the gene, and when the gene is changed, this will lead to changes in the protein, which will in the end lead to changes in the trait that is controlled by that specific gene. That mutation can create a new allele. So one more time, alleles are versions of the same gene, different versions of the same gene. Assortment of alleles. So recall, a few slides ago we've learned about meiosis, briefly talked about it, that at the beginning of meiosis a cell has two copies of each chromosome. Before the chromosomes are separated and new cells are formed, they go through a process called crossing over. So if we have a quick look, going back, on meiosis we have two sets of chromosomes here, and meiosis reduces these two sets to one set of chromosomes. Each cell usually contains, human cells usually contain 46 chromosomes, 23 times two sets of chromosomes. After meiosis, we halve this number to 23. Before these chromosomes, this chromosome number is halved to only one set of chromosomes. A process called crossing over can, or very often does, occur, where the homologous chromosomes, pairs of chromosomes, may exchange pieces with each other, and alleles might be shuffled up a little bit, which we call the assortment of alleles. Now we can see here this piece of the chromosome does exchange. The small b here goes over to that chromosome, and the big B goes over to that chromosome. So these letters represent alleles, and we have two genes here. We have the A gene and we have the B gene, and both of them have different alleles, two different alleles. We have capital A allele and lowercase a allele, and we have capital B allele and lowercase b allele. What these capital and lowercase letters directly mean, we will talk about in a moment. And so for now, we have gene A and gene B, each with two different types of alleles, one represented with a capital letter and the other represented with a lowercase letter. And we can see that during crossing over, we have a little bit of shuffling up of these alleles, and we have recombined chromosomes. So not only our genes and alleles affect our traits, how we look like and how our body behaves. Some traits are just controlled by our genes, like our blood type, for example. They're directly the result of the alleles that you have, inherited from your parents. It's simply genetically determined. If you get an A allele for your blood type from your mother and a B allele from your father, then your blood type in this case will be AB. That will not change throughout your lifetime. That is simply determined by your allele combination in the DNA of your cells. However, some other traits are the result of a combination of your alleles, of your DNA, and environmental factors. Now, the following table describes 3 examples of traits that are linked to specific genes but can be altered by the environment. We have body height, for example. Here we have several genes, not only one genes, with different alleles that influence a person's height. However, it depends as well on the diet. A diet low in needed vitamins and minerals can limit how tall a person will grow. So it depends on your genetic makeup and on your diet how tall you will grow. Handedness, for example. The handedness gene has right and left-handed alleles. A person can learn to use the opposite hand, often if his or her dominant hand is injured, for example. Are you left-handed? Are you right-handed? Did you ever injure the hand that you use most often? Maybe you broke your arm once, and then you had to use your other hand. Again, an environmental factor that can change this. Cancer, for example, is another factor, another trait. Certain alleles make a person more likely to develop certain types of cancers. However, many lifestyle choices like smoking or less exercise or a fatty diet can increase or eventually as well decrease a person's risk for certain cancers. Here we have another example of 2 identical twins. Identical twins look very, very, very similar at birth. They are genetically identical, but the older they get, the more different they might look. One of the twins may choose a healthy and low-fat food diet, whereas the other may choose an unhealthy food diet. In this case, this person, Jennifer here, her obesity genes are not triggered, whereas Karen's obesity genes are triggered. We can see a difference in the 2 individuals, although they are genetically identical. So how do these environmental effects work? This brings us to a new topic, the expression of genes. When are genes actually copied into proteins? And this is a topic that relates to the relatively new field in genetics, the field of the epigenetics. That means that genes can be activated or deactivated in cells. When a gene is actively being used to make a protein, the gene is said to be turned on or expressed. The genes for some traits are expressed all the time for sure. Others, however, are only turned on when a specific protein is needed. And that can be influenced by environmental effects. And again, the study of this field is called epigenetics. Environmental factors like temperature can also directly affect which genes are expressed. Color spots on the face, ears, feet, and tail of Siamese cats are good examples, and Siamese cats' hair on warm parts of the body stays light-colored and hair on cold parts of the body stays dark-colored. This is because the hair color gene is expressed only in areas below a certain temperature. We can see here on the right side that DNA looks different as well, where genes are turned on or are expressed compared to regions where genes are turned off. Regions of DNA on a chromosome that are turned on are loosely packed around these proteins. Now we can see we have open areas here that can easily be accessed. Now here we have turned on genes. This part of DNA is called euchromatin. Down here we have the opposite effect. The DNA is tightly rolled and packed around this protein. It is condensed, and this form of DNA is called heterochromatin. Here our genes are turned off. All right, this was part 1 on heredity. I hope I could help you understanding a little bit more about DNA, chromosomes, genes, and alleles, and how alleles affect your traits, or genes affect your traits in your body, how proteins are involved there, and how the environment can affect your traits over your lifetime. Next time in Heredity Part 2, we will have a closer look how these traits and alleles are inherited, are passed on from parents to children, and different methods to figure out probabilities of inheriting certain traits. Until then, have a great day ahead, and I hope to see you for part 2 on heredity. Goodbye.